So after you collect the sample, so for example, if you use the direct sampling method, immersive sampling method, then you have to get the animal and then remove only part of the tissue, because you don't really need the whole animal. So you might need only some of the tissue, so you can collect the tissue while the animal is alive or after is dead as soon as possible. Because after the animal die, the DNA of the tissue will degrade very fast. So you have to make sure you get the enough tissue so that you can obtain sufficient DNA. So for most of the DNA extraction protocol and the downstream lab procedure, you only need small amount of the tissue. So for example, this is one of the extraction kit, okay, extraction method. So this is the amount of the starting material, so the tissue that require for you to get a certain amount of the DNA. So you can see for different type of tissue, you need different amounts, and the quantity of the DNA also differ. So for small organism, usually we will use the entire animal or the whole plant. So for large organism, we will just take a piece of tissue. So you have to select the part of the plant or animal that are relatively free from the compound that can damage the DNA, and the compound that can inhibit the subsequent molecular work, for example, the PCR, and also the foreign DNA. So for the plant, usually we select the young or actively growing leaf. Okay, so for the plant, we usually take this part. But other parts are okay as well, but if possible, you try to choose the part that is young and actively growing. For animal, so you can use any type of tissue. For insect, okay, so usually you will take a piece of the leg, okay, here, this part, where inside the skeleton, there are a lot of the muscle tissue. So this part is rich in DNA. Or we can get the muscular tissue from the wing. And for the butterfly, you can get the DNA from the thorax or head. So there are few important things you have to remember when you remove the tissue. You have to make sure all the scalpels, forceps, vials, and container, all the material and apparatus that you use for when you try to cut a piece of tissue from the animal, okay, sterile. Okay. You also need to make sure you weigh all the curve so your DNA will not contaminate the sample.